Xtalks is the industry leader in connecting life science, food, healthcare, and medical device professionals. The Xtalks Life Science podcast features insightful conversations with industry experts about all things pharma, biotech, and more.
Hello and welcome to the Xtalks Life Science Podcast. I'm Ayesha Rashid, Senior Life Science Journalist at Xtalks.com, and this week I'm delighted to introduce a very special guest on the show. We have with us today Dr. Karen Ho, who is the Vice President, Translational Medicine at Clene Nanomedicine, which is a biopharmaceutical company focused on developing treatments for neurodegenerative diseases to restore and protect neuronal health and function. Dr. Ho is responsible for translating preclinical successes of Clene's lead drug assets into high medical value with applicability to multiple disease areas. Dr. Ho has a PhD in Developmental Biology from Stanford and completed her postdoctoral training as a National Sleep Foundation Pickwick Scholar and Howard Hughes Medical Institute postdoctoral fellow at University of Pennsylvania in the Department of Neuroscience. Dr. Ho serves on several rare disease group scientific advisory boards and holds a concurrent position as adjunct faculty at the University of Utah School of Medicine in the Department of Pediatrics, Division of Medical Genetics. Clene Nanomedicine is developing nanotherapeutics that target cellular energy impairments common to neurodegenerative and many other diseases. Specifically, the company's lead asset is based on leveraging the catalytic therapeutic activities of gold when engineered as clean-surfaced faceted nanocrystals. To learn more about the company's Nanocrystal therapeutic program, let's turn to Dr. Ho now. Dr. Ho, thank you so much for being on the show.
Thank you so much for having me.
So I'd like to ask you about your journey really from academia. You were at Stanford, which has a very strong academia industry kind of startup sort of a focus. And I'd really like to know about, how those experiences, how that experience there shaped your journey from academia to industry now, and again, how those experiences at Stanford as well as UPenn shaped your approach in translational medicine.
Sure. So my journey to Clene was actually shaped by a personal event rather than my training, but I will mention some of my training as well. Up to the point in time of this personal event, I had enjoyed a fair amount of career success. I had been admitted to Washington University in St. Louis to study biochemistry. I graduated there with a bachelor's degree in biochemistry, summa cum laude. I was then tapped as a Marshall Scholar and was able to study in England for 2 years at Cambridge University, where I studied the evolution of the arthropod body plan with a wonderful mentor and scholar, Michael Akam. I then came back to the United States for my PhD, my graduate work in Stanford, where I got a PhD in developmental biology, and then finally ended up at UPenn as a postdoc studying neuroscience, again in fruit flies, studying sleep and circadian rhythms in fruit flies. And of course, my goal was to become an academic researcher and share the beauty of biology with everyone. But then something unexpected happened. In graduate school, I met and married my husband, Suresh Venkatesubramanian, and we were expecting our first child when I was a postdoc at UPenn. And everything was going fine in the pregnancy, but at 32 weeks, he came early, and it was kind of a traumatic birth because I had I did have a placental abruption, which is a medically challenging condition in which both the mother and the baby, can lose their lives. And it turns out that I have a genetic predisposition to preeclampsia, which is when the pregnant woman's high blood pressure rises quite significantly, and that can endanger the baby and actually have to have an emergency C-section. And so the baby was born early. And I was in the NICU, unable to touch this baby because his skin was too fragile to be touched, thinking, all my life I've studied biology and I've made biology my career, but I have failed this baby because I can't do anything to help him. And that's what changed me. That's when I thought, okay, I've been studying fruit flies all this time. Maybe I should apply my biological knowledge to trying to understand how to help human beings. And on top of that, because my baby ended up being just fine after several harrowing events during his NICU stay, I had a deep gratitude for medical science that I did not have before. And so those two factors combined, my appreciation for medical science and my desire to make a difference with the power of science, actually propelled me toward the journey that I'm on now, which was in a drug development company trying to bring about treatments and cures for people who are suffering from some intractable and very difficult to treat diseases.
Wow. Dr. Ho, thank you so much for sharing that very personal story. And that, I mean, I can't even imagine how difficult of a time that must have been, and for you to have been a warrior through it. And I'm so happy to hear that your son is well, and that really puts so much into perspective, really. And it's so wonderful to learn how that very traumatic event shaped your subsequent journey into the field, and why you're doing what you're doing. Thank you so much for that. So, I'd like to now ask about Clene Nanomedicine and the fantastic work that you're leading there. And I know that the company is focused on neurodegenerative diseases and other areas as well. So, but what is the overarching sort of mission and goals of the company?
So our mission is to revolutionize the way neurodegenerative diseases are treated. We want to really positively impact the lives of millions of people who are affected by these diseases. And we're doing that by inventing a new class of drugs that unite concepts from chemistry, physics, material science, and applying that to biology. So what we're trying to do is something very new. And we think that there's a need for new ideas in treating neurodegenerative diseases. Our lead asset is called CNM-AU8, and it is being developed now to treat amyotrophic lateral sclerosis, also known as Lou Gehrig's disease, multiple sclerosis, and Parkinson's disease. So our main area of research involves the development of innovative catalytically active drugs to treat neurodegenerative disease in an entirely new way.
That's fantastic. And I'm very excited to learn more about your lead candidate and really leveraging the power of gold nanocrystals, which for me it's so innovative, because it's not every day in drug development that you hear about the use of gold nanocrystals. And so could you talk to us a bit more about this very innovative and exciting technology?
Yes, of course. In fact, that's one of the challenges that Clene faces, is we have this very, very new technology that on first glance seems a little bit wild, and in fact seemed a little bit wild to me before I first joined the company. And then when you start to see the outcome of treating with this drug, you start to believe that there is something very interesting and very real here. So in the background is actually a cover of one of the journals in which we published. That is a nanocrystal that has been rendered for us by an artist. That nanocrystal is active because it is actually catalytically active. Now, we know that we have treasured gold for centuries, because it's not chemically reactive. It's not corrosive. It doesn't rust. It doesn't react with its environment. That's why it stays shiny. But it turns out that if you take gold and make it into the nanocrystal form, so we're talking about crystals that are smaller than a red blood cell, smaller than a mitochondria inside of living cells, now we have a way of aligning those atoms of gold in a crystal shape that's kind of like a faceted diamond. And once you make the gold nanocrystals like that, they become highly catalytically active, meaning that they are chemical reagents. They can interact with chemicals in their environment. And what we found is that these gold nanocrystals actually can take a very important energetic metabolite that's located in all living cells and convert it into something that is energetically useful to diseased and dying cells and convert it actually into ATP, the currency of all energy. And so that's essentially what these little nanocrystals do. We actually can drink them. They're in this water suspension. So we drink them and then they go into our gastrointestinal system. They get it out into the bloodstream and then they can get through the blood brain barrier into the brain where they can get inside of diseased cells. And we know that dying cells that are associated with neurodegenerative diseases are fighting back against a lot of different stressors. They're fighting against oxidative stress, they're fighting against autophagy stress, the mitochondria are dysfunctional. And so they have all of these stressors. And in order to fight back against those stressors, they need energy. And they consume a lot of ATP in order to do this. So you can kind of think of these little gold nanocrystals like little mitochondria that are producing more energy for the cell free of charge so that the energy can now be used to remyelinate, to neuroprotect, to help that neuron function and fire and do all the things that neurons do.
Wow, that is so neat, I would say, to, come upon this kind of a discovery where you can take gold and develop it in a way where you have these nanocrystals and becoming sort of a powerhouse akin to mitochondria. That is so innovative and so cool. So, wow. And so in terms of the lead candidate that Clene has, what indications are you investigating them in?
Yeah, so we are currently focused on amyotrophic lateral sclerosis,
Right, you mentioned,
multiple
yes.
sclerosis,
Mm-hmm.
and Parkinson's disease.
Okay, wonderful. And so, what is it? Is there something particular about how these gold nanocrystals can specifically target neurodegenerative diseases? I know you mentioned that they can help in restoring, providing the energy needed to restore, let's say, myelination of neurons. But is there something specific to these specific subset of diseases where this nanocrystal technology really works?
That is an excellent question, Ayesha. In fact, we feel and we believe that our evidence shows that there are a number of different types of neurodegenerative diseases that CNM-Au8 could treat. And that's one of the exciting things about it because here we have a catalyst. And when I say catalyst, that means it has to take a reagent from its environment, a chemical from its environment. In this case, it's nicotinamide adenine dinucleotide hydride, or NADH, that energy metabolite that I was describing earlier. And it can efficiently convert that into NAD+, which is essentially what mitochondrial complex 1 does. It takes NADH, converts it to NAD+. And then that drives the ATP production in mitochondria. Similar things are happening with CNM-Au8. But what is also very interesting are these indications all share a common disease feature. And what we know about them is that they present very differently in the clinic. It's very easy to distinguish someone who has Parkinson's disease from someone who has multiple sclerosis. However, what is underlying the disease state is very similar across multiple diseases. And why is that? That's because neurons in the brain are one of the most highly energetically demanding cells in the body because of all the different functions that they have to perform. So when you have even a slight insult on the energy production inside of a neuron, that neuron's going to suffer a lot because it's so dependent on energy in order to function. So what we've found is that multiple sclerosis, amyotrophic lateral sclerosis, Parkinson's disease, and in fact, a lot of other diseases all share the common phenotype that at some point in their disease state, mitochondrial function is compromised. And when that mitochondrial function is compromised, now we can go in there, treat with these gold nanocrystals, help with the energy production, and help possibly reverse some of the disease states that are now cascading toward neuronal death.
That makes so much sense in terms of really hitting the underlying mechanism of action of the pathogenesis of the diseases which are in common. So the mitochondrial dysfunction here. So that's fantastic. That's definitely essentially what a targeted therapy is, and using these nanocrystals to really target the underlying pathogenesis of these diseases, I think is really key, of course. So that's really, really cool to learn about. And in terms of it kind of boggles my mind thinking about, nanocrystals, just given their scale and how you're able to take gold and develop these nanocrystals from them. And so what does that entail from a production aspect? How difficult and challenging is it to develop these kinds of nanocrystals? And how does the electrocrystal chemical production process pan out and how is it different from traditional drug formulations?
Exactly. Great question. So the way we make our gold nanoparticles differs significantly from the way gold nanoparticles are traditionally made. You probably have heard that gold nanoparticles are used in medical applications and in imaging all the time.
Yeah.
But it turns out this traditional method involves chemical reactions in which gold salts are reduced to make gold particles using organic chemicals. These organic molecules get stuck on the surface of the gold nanoparticles, covalently bonded, in fact, and this changes the properties of those gold nanoparticles. Now, one aspect of the secret sauce of CNM-Au8 is that the surface of the gold nanocrystal has to be clean in order for it to act as a catalyst, because gold nanocrystals are actually surface catalysts. So if we think of enzymes as catalysts, enzymes have an active site where they bind that active site, and that's where the change it takes place, where the chemical reaction takes place. Well, it turns out that the catalyst, the active site for these catalysts is their surface. So now if you stuck organic chemicals onto the surface, there's very little to no catalytic activity, which is possibly why we missed gold nanocatalyst activity for a long time in scientific endeavors. In addition, the surfaces should be clean because the organic solvents and stabilizers actually change the toxic properties of the gold nanoparticles. If you go and look in the literature, there will be lots of articles showing, gold nanoparticles are toxic to this organism. They kill these cells. They cause reactive oxidative stress in the brain of these rats. And that's because there are these organic chemicals on the surface of the gold nanoparticles that change their properties. So we invented a way, and by we, I should say, it's Mark Mortensen, our CSO, as well as his team in our lab in Maryland, who invented a way of using clean electrocrystal chemistry to make stable gold nanoparticles that are crystalline form, that are that have clean surfaces, and are highly faceted so that they have this catalytic activity in extraordinary high catalytic activity. And now we are in a position to take the gold nanocrystals that are made using this novel production method into the living organism and explore the benefits of how having highly catalytically active gold nanoparticles with low toxicity can have impact on cells.
Absolutely. And so I know that your team at Clene recently published findings on the protein corona composition of gold of the gold nanocatalysts, nanocrystals. And so could you explain some of those findings and also explain the significance, again, for the treatment of neurodegenerative diseases?
Of course, sure. So, this was a publication that came out in ACS Pharmacology and Translational Science. And it was a wonderful collaboration that we had with Professor Morteza Mahmoudi and his lab at Michigan State. What we knew about gold nanoparticles going into this work is that all gold nanoparticles, because their surfaces are so chemically reactive, they like to bind proteins. And what happens is whenever they interact with an environment that is protein rich, a protein corona or a shell will form around each nanoparticle. And the protein shell for each nanoparticle can be different depending on the composition of that nanoparticle. So in this case, it's gold, the size and shape of that nanoparticle. And so what we wanted to do was ask the question, when our gold nanocrystals come into contact with human blood, and in this case we use plasma from blood, which is protein rich, what kinds of proteins are attracted to their surfaces because we have a unique gold nanocrystal? Um, and we wanted to know what was the nature of those proteins. So we teamed up with one of the lead experts in this field, Morteza, and he and his lab took our gold nanocrystals, mixed them with plasma, and asked what proteins are actually stuck to the surfaces. And what we found was very rewarding, actually, was that these proteins that are enriched on the surfaces of our gold happen to be apolipoproteins. Well, what we know about apolipoproteins is that they naturally can cross over the blood-brain barrier. And we also know that when they are bound to other things, they can act as shuttles, taking whatever they're bound to across the blood-brain barrier as well. So that made perfect sense with some of our other studies in which we asked patients with Parkinson's disease or multiple sclerosis to drink some of our gold, and then we measured changes in their brain. So our gold nanocrystals, when drunk, was getting into their brain and changing the brain metabolites, energy metabolites in the brain in a favorable way. And that transversal of the blood-brain barrier made sense because of the characterization of these proteins that can also shuttle gold nanocrystals across the blood-brain barrier.
That is very fascinating. Really, what's the word I'm looking for? Very elegant. I mean, gold is always known to be elegant jewelry, but in this scenario, in this context of developing it as a therapeutic, I mean, this is such fascinating stuff. And I am so happy to be learning so much about like the possibilities of gold and just imagining it as a drug one day. This is so, so cool. And in terms of your lead therapeutic candidate, what stage of development is it at in the various indications that you're working in?
Right. So right now we're on the cusp of a phase 3 clinical trial for CNM-Au8 in ALS, as well as in multiple sclerosis. And That's really where we are right now. Because what we've shown with our phase 2 clinical studies in ALS, for example, is that we can decrease the risk of death in ALS patients. This is significant because ALS patients, once they are diagnosed, have on average 2 to 5 years to live. And so what we've seen is in 2 independent phase 2 clinical trials, a resounding signal of the extension of life with patients treated with CNM-Au8. In addition, in multiple sclerosis, we looked at something different. We wanted to see evidence of remyelination. And so we admitted into our clinical trials individuals who had damage to their optic nerves through visual impairment. And we measured the increase in their vision as a way of asking whether or not we were neuroprotecting and remyelinating, possibly remyelinating. And that's what we saw was a very extensive increase in their low contrast letter acuity. That is their ability to see letters on a typical eye chart that had been grayed out. So they're actually pretty hard to see. And what we found was that, in fact, over the long term extension of the study, patients treated with CNM-Au8 over several months to years actually improved their score on low contrast letter acuity. And they improved, and this was kind of a surprise to us, improved their cognition, which was measured by a test called the SDMT, which is a test of working memory. So that's a pretty exciting future for us.
Absolutely. And with taking those very promising results, what are some of the key milestones that we can look forward to from Clene in the coming months and the next couple of years?
We would very much like to get CNM-Au8 approved for ALS and multiple sclerosis. And those 2 approvals will be specifically significant because it will show that you can use catalytically active nanocrystals to treat these very hard to treat neurodegenerative diseases. And so it will kind of open up a field of new ways of exploring how we can use these principles that we've tried to harness from chemistry and physics and material science in better ways and more effective ways for even more diseases. And so I think those would be the next 2 immediate steps, getting the approval, and then beyond that, developing a field for new kinds of therapeutics.
Indeed. And you did mention that leveraging this technology for other diseases. What kinds of diseases do you think that this could be translated over to?
Well, I would love to see us then reach into pediatric rare diseases because one of the ways I got into drug development was my understanding of the great need that forgotten diseases, that rare diseases have faced in the past, including ALS. And it is these diseases that we really can start to understand and treat with catalytic nanocrystals, as well as genetic therapies, as well as all of the many exciting new technologies that we're just on the cusp of bringing into fruition. So really for CNM-Au8, mitochondrial diseases, pediatric rare diseases, all of which have some aspect of mitochondrial dysfunction, I think would be very, very good new indications to go after. And in fact, last month I presented a poster at a Rett syndrome meeting showing that CNM-Au8 has some promising preclinical preliminary results for the treatment of Rett syndrome, which is a syndrome that affects children and can cause seizures and developmental delay, and is a disease that is in need of some treatments.
And so I'd like to ask, in really pioneering novel technologies, novel drugs, based on, again, very new technologies like gold nanocrystals. Just taking a step back, like, what do you think is key in really working in this kind of space with such novel technologies? With so many unknowns, like what would you, I would say, what kind of advice would you give to people, embarking on, new territory like this? And what are the skills and what are sort of the perspectives that you need?
Well, I think that we are all natural scientists and we are all deeply curious people. And I think that we need to keep that spirit of curiosity very close at hand. There were two very important things that I learned from my training in developmental biology, genetics at Stanford and at UPenn. And that is that you can start with very little knowledge about a field and learn things quickly enough to start to make contributions. That is what graduate school is really all about. And so anyone who has gone through a training process that is similar to graduate school, where you start off knowing very little and work very hard and start to become an expert in a very tiny part, but a significant part of a field, is a skill that can be applied to any aspect of technology and scientific development that we have in this world. And so that's a very important skill to have. And I think that even people who decide to not go into science after graduate school can really benefit from knowing that there is value in pursuing that kind of trial by fire. The other thing that I have learned, and that is from working for a small company where I've had to wear many different hats and have many different responsibilities, is that we need to use every tool in our knowledge toolbox to tackle hard problems. And so developmental biology is the study of how a fertilized single cell becomes a multicellular, fully formed, functioning adult. And it was from that training in Stanford where I learned you can use your principles from chemistry and physics and cell biology and structural biology to try to answer some fundamental questions about how a fertilized egg becomes a fully functioning adult. It's one of the most beautiful questions in science, I think, and still is. But using every tool in your scientific toolbox is a very important principle. So I'm just very grateful that we have had so many different tools that were required to get in there and start to figure out how CNM-Au8, for example, is working from many different angles, which is what we're doing and continuing to do at Clene.
Well, thank you so much for sharing your story and the story of Clene and this very innovative technology that the company is developing under your leadership. And thank you so much for the work that you do. And we're at Xtalks, we're definitely going to be keeping an eye out for all of the latest developments from Clene, and hope to really, hope to have you back and to share the next milestones and successes. So, Dr. Ho, thank you so much for your time today.
Thank you so much, Ayesha. I really appreciate it.
That's the end of this episode of the Xtalks Life Science Podcast. If you liked today's show, don't forget to rate, review, and subscribe. Thank you, everyone, and we'll see you next week. Bye for now.
To join the Xtalks community, visit xtalks.com and register for exclusive access to curated content like live webinars, news articles, and educational videos. The views and opinions expressed in this podcast are those of the speakers sharing them and should not be taken as professional advice, nor do they necessarily reflect the policy or position of Honeycomb Worldwide. For further information, email us at podcast@xtalks.com.